Hello everyone. So welcome to another topic of biochemistry nutrition. Today we are going to read one of the vitamin B complexes, that is vitamin B12. So the vitamin B12, the another name of vitamin B12 is cobalamin. Now let us discuss about various sources of vitamin B12. One of the good sources of vitamin B12 is curd, and mainly the vitamin B12 is found from the animal sources that are meat, fish, poultry, shellfish, milk, cheese, and egg. The vitamin B12 is present in negligible amount, or it is not present in the vegetable sources. So there is more chances of vitamin B12 deficiency in the vegetarian people. Now let us discuss about the recommended daily allowance of vitamin B12. It is 1 to 2 microgram per day, whereas in the pregnancy it is more than 2 microgram per day. So as I have said that vitamin B complexes are the water soluble vitamin, and it is washed away due to it is soluble in water and it is excreted through kidney, and it is not stored. Vitamin B and vitamin C, mainly water soluble vitamins, are not stored, but the vitamin B12 is the exception. The vitamin B12 is stored in the body. So one of the exception of the water soluble vitamin is vitamin B12 is stored in the body. There is large reserve pool that is stored in the liver. The vitamin B12 is synthesized by the microorganisms too. Now let us discuss about the various functions or physiological actions of the vitamin B12. The vitamin B12 acts as cofactor for enzyme methionine synthase that transfers CH3 group as methylcobalamin, and it also helps to the enzyme methylmalonyl CoA mutase. How it helps, I will discuss this in detail. So as you can see in this picture, I'm just going to explain this in brief. So in the subsequent lecture in the biochemistry metabolism, I will discuss it in detail. From the protein, the methionine is obtained, or we can say that from protein methionine can be converted, and from the methionine it can be converted to protein. So from the methionine, as we can see, from the methionine, the methionine is converted to S-adenosyl methionine, S-adenosyl methionine. From the S-adenosyl methionine, it is converted to S-adenosyl homocysteine, and from the S-adenosyl homocysteine, it is converted to homocysteine, and from the homocysteine is again converted to methionine. So while conversion of the homocysteine to methionine, in this cycle there is requirement of vitamin B12 and vitamin B6. The large proportion of vitamin B12 is required, whereas a small proportion of vitamin B6 is required here. So during the formation of the tetrahydrofolic acid also vitamin B12 is required while transferring from while the homocysteine is converted to methionine. The vitamin B12 is required for the making of tetrahydrofolate. So there is the main role of vitamin B12. Now let us discuss the another, that is methylmalonyl coenzyme A and succinyl coenzyme A. The role of methylmalonyl coenzyme A mutase, the interrelationship between vitamin B12 and methylmalonyl coenzyme A mutase, as I have discussed here. So fatty acid with odd number of carbons or branch chain amino acid is converted to methylmalonyl coenzyme A in the various steps. So the methylmalonyl coenzyme A is changed to succinyl coenzyme A by the action of the enzyme methylmalonyl coenzyme A mutase under the cofactor of vitamin B12. So the conversion of methylmalonyl coenzyme A to succinyl coenzyme A requires the cofactor that is vitamin B12. So without vitamin B12, there is no successful conversion of methylmalonyl coenzyme A to succinyl coenzyme A. And from succinyl coenzyme A, the cofactor vitamin B6 is required to convert to heme, and succinyl coenzyme A is also converted to tricyclic acids, TCA. So as I have already discussed, the vitamin B12 acts as the cofactor for enzyme methionine synthase and methylmalonyl coenzyme A mutase. So it is very important for DNA synthesis in the mainly two reaction. Again, I am going to say to you, synthesis of methionine from homocysteine and isomerization of methylmalonyl coenzyme A to succinyl coenzyme A, as I have discussed this in two pictures here subsequently. So this is the main role of main physiological functions or physiological actions of the vitamin B12 in DNA synthesis, and these two important reactions for conversion of the homocysteine to methionine and methylmalonyl coenzyme A to succinyl coenzyme A. Now let us discuss the various causes for the deficiency of vitamin B12 and what are the various manifestations that occurs when there is deficiency of vitamin B12. Now moving towards the deficiency of vitamin B12, let us discuss first about what are the various causes of the deficiency of vitamin B12. So first is nutritional deficiency. So as we all know that the rich sources of vitamin B12 are animal products. So those who are strictly vegetarian, they have more tendency to manifest the vitamin B12 deficiency syndrome, or they have more chances of vitamin B12 deficiency. And another is malabsorption. So the decreased absorption can take place in the lack of intrinsic factor, like in pernicious anemia, and in the surgery, like there is absence of terminal ileum due to surgical resection. So another is pre-enteritis or achlorhydria. As we all know that the absorption of the vitamin B12 requires the hydrochloric acid and intrinsic factor. So another one is there could be the increased requirement of vitamin B12 condition like in pregnancy, or there could be the infestation of fish tapeworm. If there is infestation of fish tapeworm, the person may be perfectly fine. There could be the adequate nutritional intake, but the requirement will be more because there will be infestation by the fish tapeworm. So now let us discuss what are the various manifestations of the vitamin B12. The various hematological manifestations of the vitamin B12 are macrocytic anemia, that is megaloblastic anemia, and hypersegmented polymorphonuclear cells. And also there will be the various neurological symptoms that occurs due to demyelination like paresthesias or subacute combined degeneration. In the subacute combined degeneration, what happens is that there will be the degeneration of the dorsal column, degeneration of the lateral corticospinal tract, and there will be degeneration of the spinocerebellar tract. So that is the subacute combined degeneration. So in the lab value, what we can see is that there will be the increased serum homocysteine and there will be the increased methylmalonic acid levels in the serum, along with the secondary folate deficiency. So if there is prolonged vitamin B12 deficiency, then there will be the irreversible nerve damage. The various neurological symptoms will be permanent. So the anti-intrinsic factor in the serum also confirms the diagnosis of the pernicious anemia. But if we supplement only folate, the folate supplementation can mask the hematological symptom but not the neurological symptom, because if there will be the folate deficiency, then the only hematological symptom occurs, not the neurological, but both hematological and neurological symptom occur in the vitamin B12 deficiency. So now this was for today. I hope you understood this lecture. Thank you.